Welcome to Professor SNAB, where we make A-level biology as simple as a cell. So today we're understanding the roles of antigens and antibodies in the body's immune response, including the involvement of plasma cells, macrophages, and antigen-presenting cells. So, what are antigens? You might have briefly explored these in GCSE, and these are the markers found on cells, bacteria, and viruses. And antigens are unique to each cell type or microorganism. So in the case of cells, their entire purpose is for cell-to-cell recognition. And in some cases, the glycoproteins or the glycolipids found on the cell surface act as antigens. There are two types of antigens. So you have self-antigens and non-self-antigens. And self-antigens are produced by the organism's own body and do not cause an immune response. However, non-self are not produced by the organism's own body and will lead to an immune response. So, antigens and phagocytosis. So, after the pathogen is engulfed by the phagocytosis, the antigens are presented by being passed to the cell membrane. This results in the phagocytes becoming antigen-presenting cells. Macrophages activate the specific immune response as appropriate in case of a future attack. So they do remember the antigens. So the whole idea of antigen becoming antigen presenting cells is that they remember the antigens for the future, so that they can attack in case that a pathogen enters the body again. So just to clarify, macrophages, they are a type of phagocytes that are involved in remembering. So they have that special feature. It's not something you need to specifically know, but it's just extra knowledge. So yeah, antibody structure. You need to know this. So keep an eye on the slide. So antibodies are Y-shaped molecules, and they are sometimes called immunoglobulins. They consist of 4 polypeptide chains, 2 of which are heavy chains and the other 2 are light chains. The heavy chains, which are long, are attached by disulfide bonds to the light chains, which are short. So polypeptide chains consist of a constant region and a variable region. The constant region do not vary within a class of antibody. They are the same within a class of an antibody. So on the other hand, in the variable region, the amino acid sequences vary for each antibody, which means it's unique for each one. So an antigen-antibody complex is formed when an antibody binds to the antigen in the variable region. The antigen binding site is the site at the end of the variable region. The binding sites vary from antibody to antibody, making the response very specific to the antigen. The hinge region, again, you need to know this slide as well. The hinge region is where the disulfide bonds join the heavy chains, and this gives flexibility to the antibody molecule and allows the antigen binding site to be placed at different angles as necessary when binding to the antigens. The presence of the hinge region varies between the class of antibodies. So antibodies can be membrane bound or secreted directly into the blood. So those are the two types of antibodies that you can have. So membrane bound antibodies are attached to the surface of the lymphocytes, and they have an extra section of polypeptide chain within the heavy chain, and this is what allows it to attach itself to the lymphocytes. So this section, as you know, as you can guess, is not needed if it's an antibody that is secreted directly into the blood. So they can be removed using a process called alternative splicing. So splicing is a process that removes the coding sections of mRNA, which are also called exons. So over here, the code for the extra section that is not needed is removed. So antibodies first bind to the specific antigens. This triggers a specific immune response. So as pathogens enter host cells by binding to them using the receptors on the surface, antibodies can prevent pathogens from infecting host cells by binding to these receptors. So antibodies can also act as antitoxins. So you might have seen these as 3 markers in GCSE. But yeah, they can also act as antitoxins. So this is by binding to the toxins produced by these pathogens. Antibodies can cause pathogens to clump together. So this is through a process called agglutination. And this is then engulfed by phagocytes to phagocytosis, which you guys are very familiar with. So yeah, that is all from Professor Snap today. Hope you found this topic very useful. It's quite a very important topic, especially everything around the function and the structure of antibodies. And I will recommend going back to this video a couple of times to fully understand it. And of course, if you have any questions or if you need clarification, let me know in the comments below and I'll get back to them as soon as possible. And don't forget to like, share, and subscribe.